So hi, thanks very much for inviting me. Thanks to the organizers for allowing me to fulfill my life's ambition of speaking on the main Cosyne podium. I shall be retiring in about 45 minutes. When I was still working, I was mostly interested in these types of problems, which are how we control our behavior, how we learn and represent and make decisions in these kinds of complex worlds. Recently, I've been focusing with my lab on just one aspect of this problem, which is how I represent these links, the relationships between events and objects in our world. And so this is, I think, an important problem for most of cognition. It's obviously important in reward learning, where our job is to associate stimuli with rewards, for example. But in other types of cognition, we have to represent the relationships in our social world, the relationships in space if we're moving around, and even we have to piece together the different events and characters that make up a story. And so I think it's an important and general problem. And if you're interested in something abstract like that, which is how do you go about representing a link or a relationship, then it's interesting to consider what kind of representation you might go looking for. And so here's an associational relationship that many people would have experienced this morning, the relationship between this Starbucks fairy and this cup of coffee. And if you wanted to represent that relationship, one thing you might think of doing is taking some cells which represent that Starbucks fairy and some cells that you already have that represent the cup of coffee and building some kind of new excitatory connection between these cells. And I'm going to refer to that as an implicit code for a relationship. And there's plenty of evidence that this kind of code exists, some from my lab, but many other things as well. You could, I think of a different way of doing it, which I'm going to call an explicit code for a relationship. And that's to look at some separate pool of neurons that never knew anything about the Starbucks fairy or the coffee, but are coding for the fact that those two things are related in some way. And it might not immediately seem obvious why that would be profitable, but I'm going to try to persuade you that it is profitable during the course of this talk. I'm just going to show you one example right now, and then there'll be more later. But if you had a set of neurons that knew about a relationship, that coded particularly for a relationship, then if you saw another example of that relationship, like the relationship between this Nike swoosh and these boots, then you could learn about it much faster because you'd already have a representation that you could use. And so it's very useful for generalizing knowledge. Okay, so just bear with me for now and assume that these explicit relational codes exist in your brain. Then you've got a new problem, a new thing to worry about, which is when you go to Starbucks, you might want to have more than just a coffee. You might be interested in, for example, in this attractive piece of red velvet cake. And then now you have to start coding a web of interrelated objects. And so you could choose to do that by making new codes or reusing the same codes to represent independently each one of these links in this web. Or, and I'm going to call, and that would be an explicit code, but it would be local because the cells know something about each link independently. An alternative would be to, in some magical way, have a population of cells that are able to represent the structure of the whole map. And so that's what I'm going to call an explicit global code. Okay, so in this talk, I'm going to try to persuade you that relational structures in frontal cortex can be explicit. That means divorced from their sensory elements, and therefore generalizable to other sensory elements. That these structures control how new stimuli are processed. So when new elements come along, they're controlled by these abstract, explicit relational structures. I'm going to try to persuade you that if you're going to code those relationships, you better do it globally, because you can do much more stuff with them if you do. And then I'm going to try to show you some evidence from the human brain that these global relational codes exist. And I'm going to use some analogies to spatial reasoning for that. OK, great. So let's start at the beginning, and let's start really simple. This is a task that many animals have learned in many labs. A simple discrimination task. What you want to learn, that this star-shaped thing leads to reward, but this bell-shaped thing doesn't. And the way this has been trained many, many times since Pavlov is to do something like this. So just to present star, reward, bell, no reward, bell, star, reward, etc., etc. And animals are able to learn these tasks, and there's theories, there's many decades of theories explaining how they do it. But it's worth considering whether this really is a simple task for animals in labs, whether they really are doing conditioning-like things. Because what we're asking them to do is something like this. We're asking them to understand that the reason they're getting a reward is because they just saw a star. And this isn't something that comes natural to an animal in a lab. Instead, perhaps they could have thought something like this. They could have thought something completely different. They could have thought maybe the apple juice came because of the star that happened 4 trials ago, or that the bell-shaped thing happened because the apple juice happened, or something like that. The animals in the labs don't have access to the experimenter's idea about how the experiment works, and so they have to figure that out. And I think you might say, okay, they can just figure it out by temporal proximity or something, and I'll show you later that they can do that. But I think that they can do other things too. This isn't just philosophy. When you learnt about this relationship between this Starbucks and this coffee sign, you learnt it something like this. You learnt this chain of processing, but because you understood something about the structure of the world, you were able easily to make this link between the Starbucks sign and the coffee, and not make a link between the Starbucks sign and the queue for the checkout. And again, in labs, this is a problem that animals overcome. And so monkeys in Oxford do stimulus reward learning tasks often, and often they attend closely to the stimuli and the rewards, but at other times they do all sorts of other things. Here's a monkey in the middle of a stimulus reward learning task who isn't paying so much attention to the stimulus and the reward. But he was able to understand the fact that the somersault wasn't causally relevant for the reward that he was able to get later. Okay, so here's an experiment that involved that monkey, or one of his friends. But it's a bit like a associative learning task, very similar to the ones that I was showing you earlier, discrimination. But now there are 3 stimuli, and critically, there are 3 new stimuli each day. And so to start with, they're very slow at learning these rules that one stimulus is rewarding at one time and another is rewarding at another time. But they get to understand something about the task, and by the end of it, they're able to track rewards. And so I'll just fill you in quickly that one of the stimuli, the stimuli are rewarding at different times, and you can see when they were rewarding on this particular day by graphs looking a little bit like that one on the right. But critically, they learn how to learn these things because by the end of learning, they're able to track these raw probabilities very well. And so here are just 2 groups of monkeys that are able to follow, choose the best stimuli, or the best stimulus almost all the time. But we can look a little bit more carefully at how they're doing this with some detailed regression analyses. And so in these regression analyses I'm about to show you, we can ask, well, did they know the structure of the task they were being asked to perform? And if they did, that means they're going to be pairing each reward or each absence of a reward with the stimulus that occurred immediately before them, because that's the structure of the task that we imposed. And if they did that, then you're going to see high regression weights on the diagonal of this matrix. But alternatively, they could have been pairing these rewards with all sorts of other things that happened in their immediate sort of time horizon. And if they did that, then you're going to be seeing stuff on the off-diagonal terms of this regression coefficient matrix. So in control monkeys, this is what it looks like, and you can see appropriately there's a big weight on the diagonal, and I can tell you that this is a logarithmic plot, which means that big weight on the diagonal, in fact, really dominates all of the explanatory variance in the data. And so these guys have learned how to do the task. These guys are able to pair the reward with the stimulus that came immediately before it, and therefore adjust their behavior appropriately. But then if we go and make a lesion to the orbitofrontal cortex, then you can see a rather different pattern of behavior, where there's still lots of ancillary learning about what happened recently, but there's no preferential learning for the thing that happened just immediately before. And so what's happening here is that control monkeys have a representation of the task and are able to pair the reward appropriately with the thing that happened immediately before it. But OFC lesion monkeys are effectively doing just temporal correlations. They're just pairing the reward with anything happened around that time. Cool. So I hope you'll believe me then, and that's just one of many examples I could have picked, that frontal cortex is able to extract abstract rules or relationships which can then be imposed back onto new elements or stimuli. And now I'm going to try to show you that once those abstract relational structures have been imposed, they can affect how new stimuli are processed. And this is the work of Yunze Liu and Zeb Kurth-Nelson. And I'm going to try and do this by borrowing a trick from rodent tasks. This is a figure from Foster Lab, from a beautiful paper showing something called hippocampal replay. And I'm sure everybody in this audience knows more than I do about hippocampal replay, but I'll just take you through it in case there's anyone who hasn't seen it. So if you record while a rodent runs down a track or in an environment, you can decode from the hippocampal cells where the monkey is, where the rodent is. Sorry, I wish we could do it with monkeys, not yet. So you can do that, and then you can plot here. He's running down a linear track, and you can plot where you think he is based on his hippocampal cells, and he is indeed in that place. But before and after he ran down the track, you could record something a bit amazing when he was resting, which is the same sequence of cells, you can decode the same sequence of positions from that cells in a really rapid time period while he's resting. And you can see that sequence can progress either forwards or backwards, and it's much, much faster than the time it took the animal to run down the track. And what Ambrose and Foster showed in this lovely paper was that if you put a reward at the end of the track, that increases the frequency of reverse replays, but not the frequency of forward ones. Okay, so we're going to try and borrow this trick and use it in humans now. But what we're going to do is we're going to try to impose some structural knowledge, some relational knowledge on top of replay. And so we're going to ask, can this structural knowledge, can this impact on the replay that's going to happen when new learning happens on new elements? And so we're going to do that by taking a sequence, which is a bit like that Starbucks to coffee sequence I showed you earlier, jumbling it up into a wrong order, but teaching subjects how to unjumble it, like this. And then the next day, we're going to give them new stimuli, which are also jumbled up, and we're going to measure these replay sequences. I'll show you how we do that in a minute. And we're going to ask, is that replay going to replay the jumbled sequences that they've actually seen, or the unjumbled sequences that is implied by the relational knowledge we taught them the day before? That's the kind of question we're going to ask. I'll show you how this works. And so this is going to be done in a MEG scanner, and we're not going to be recording hippocampal cells. We're going to be recording the topography of on the scalp of magnetic fields. But you can do this. As shown, but there's a couple of other papers showing that you can do this really nicely. You can decode. The subjects go into the scanner. They're shown a bunch of different stimuli, and each one of those stimulus will have a unique pattern of sensors, so that you can decode from the MEG sensor pattern what the subject is looking at now. And so we're going to go and do that. We're going to go and measure these MEG sensor patterns, just like you would go and measure the hippocampal place codes, which cell is pointing to which place. And we'll get a vector of MEG sensors which tells you what the subject's looking at. And then we're going to show them two different sequences that are jumbled together, and each sequence is jumbled up in the wrong order. And then we're going to let them rest. And during this rest, we're going to ask, do those MEG sensors, do they recapitulate the same patterns that we measured during the localizer when we were recording at the start? And if they do they play them in any particular order? Do they go lion, seat, blah, 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 in a particular order? And is that order the order of the jumbled sequences that they saw, or is it the order of the unjumbled sequences that they should use? And so here's the answer. I'm just going to show you how to read this graph. Firstly, you can see that during rest, the MEG sensor patterns do recapitulate the stimuli, and they do so in sequences. And you can see that because this measure is high. It's above the dashed line, which is the multiple comparisons corrected permutation testing equivalent. And you can see that this replay is really quick. And so the time period on average between playing one pattern and the next is order 50 milliseconds. So that's looking like much, much faster replay, just like in the hippocampus. You can see that the replay goes forwards, and you can see that because this graph is pointing up, not down. And I can tell you that in order to define forwards in this analysis, we had to use the unjumbled sequences, not the jumbled ones. So if you look at the jumbled ones, remember these are the ones they actually saw, then there's no replay at all of those, but the unjumbled ones replay. And so this learnt structure that they learnt the day before is unjumbling the replay for the new stimuli. So then we can go back and play that same trick that Ambrose and Foster played, and we can reward one of those two sequences. And so we can say, at the end of this sequence, this last one, D, there's going to be a reward. And then when we do that, we can measure exactly the same thing during the next rest period, and you can measure, and you can see what happens here. The replay swaps from going forwards to going backwards. So now it's replaying DCBA, DCBA. And that backwards replay is selective for the rewarded sequence. So there's two sequences here. The unrewarded sequence is still, if anything, replaying forwards. So now during that rest period there, after the reward, you have one sequence playing DCBA and another sequence playing ABCD. Okay, and again, there's nothing for those jumbled sequences, the one they actually saw. It's all, the only thing you can measure is to do with the unjumbled sequences that have had this structure imposed on them. Okay, and just lastly, you can ask them to make a decision. So now you're asking about the two A's. You're asking, do you want this first element in the sequence or this other first element in the sequence. And obviously, they know which ones are rewarded because you've just told them. So they should be able to just choose it immediately. And indeed, they can. But when you ask them which A they want, you can measure this immediate back replay. And so it goes, which one do you want? Reward DCB, that A, that one, please. And so that's what's happening in those MEG sensors then. Cool. So there's backwards replay from the reward to make the right choice. So overall in that study, we showed them something like this, a really jumbled up structure, and then they replayed something like a sequence in the correct order. And you can see, if you want to know the details of how all these analyses work, you can see this tonight in poster 285 that Jens is presenting. Okay, so hopefully I've persuaded you that there's some abstract relational information coded in the frontal cortex or related structures that is divorced from sensory elements, and then when new sensory elements come in, how they're processed is affected by these abstract relational codes. And so, but what I haven't told you anything about is how these global relational codes might look. What might they look like? How these relational codes look. And so now I'm just going to show you a little bit of maths because it's Cosyne to try to persuade you that you want to have global codes and not local codes. And so here's how you might think about that rat running down a linear track. You might think that he has representations of different places. Certainly if you've recorded place cells, you might think that. Or if you'd modeled anything in a reinforcement learning world, you might think he has different states and therefore different elements on this linear track. But if you want to know how he can control himself in this world, you need to know something else apart from this. You need to know this, which is the adjacency matrix, which tells him which one of these place cells is next to which of the other one of those place cells. And so if you have that matrix and you have those place cells, then you can move around this world. And so here's this rat trying to move one state around this world, and to do so, he can just multiply himself by this adjacency matrix. And if he wanted to move 2 states, then he could multiply himself again by this adjacency matrix, or 3 states, he can multiply it 3 times by this adjacency matrix. And so this is Cosyne, and so everybody will immediately be saying, oh, look, all of those matrices A squared, and A cubed have the same eigenvectors. Presumably that was what was in everybody's mind at the time. And so because they do, that means that if you code for the eigenvectors or something close to them, then you don't need to go A and then multiply again and multiply again. You can jump to anywhere around the world by combining the eigenvectors in different ways. Okay, so just as an example, if you were to hold all the information of those in that adjacency matrix on the left in independent elements, we would call that a local code. It only knows about the neighbors, and you have, in order to work with that type of representation, you have to simulate going through each one of the neighbors to get to where you want to go. If you have a global code, like, for example, the eigenstructure of this entire map, then you have a code where each element doesn't know everything about one relationship, but it knows something about all of the relationships. And if you do it correctly, you can know about the whole structure at once, and you can make easy algorithms that let you jump from anywhere to anywhere else in your world. And there's been a stunning paper recently by Kim Steckenfeld, who's here, I think, showing all sorts of fun you can have with this. It was in Nature Neuroscience a couple of months ago. I'll show you one bit of fun you can have with this. This is just one example of what you can do. This is, again, that local code, but I'm showing you above that I've made it by recombining the global code, which are the eigenvectors here, V, according to the typical eigen decomposition. But if instead I had recombined them using just a different weighting, one different weight per eigenvector, I would have got a matrix that looks like this. And this matrix does cool things, right? So in this top bar, it looks like this. So it gets bigger as you get further away from your current state. And in this middle bar here, it looks like this. And so what it's coding for is the distances between all points in the world. And so this matrix has global information. It knows how far away you are from everything in your world. And you can play lots of games with that. Here's a silly game you can play. For example, you can do navigation to a reward without ever doing any forward planning. You can just use local knowledge with these global relational codes, and you can solve global problems locally. Cool. All right. And then again, I'll just give another buzz for Kim's workshop. I think it'll be really fun if anyone wants to go to model-based cognition. Okay, so just a summary for the sort of more mathematical part of this talk, which wasn't very mathematical. Because it's global, if you code these relationships globally and explicitly, because it's global, you can easily jump around maps. You can find optimal paths without searching more than one step ahead. And there are other tricks you can do. You can readily benefit from hierarchical information, for example. And then the other theoretical advantage, I think, is because it's explicit. Because it's explicit, you can generalize between problems with similar structures, even when all the elements are different, like the Starbucks to coffee versus the Nike swoosh to boots example I had earlier. I'm not going to take you through any of the maths of that because it's complicated, but if you want to see a deep net trying to solve this problem with a variation autoencoder, you can go to poster 245 tonight to meet these two young lovely chaps here, James Whittington and Tim Muller. Cool. Okay. So lastly, I wanted to try to persuade you that given we think it's advantageous to have these global explicit relational codes, I wondered if we could find some evidence for their existence in the brain. Okay, and so I think that we all know that they're there in the brain already. They're just hiding in plain sight. So here are some grid cells that we all know about since they're before and since they're awarded the Nobel Prize. And so these cells are hanging around the entorhinal cortex, just next to the hippocampus, and they do just what I was talking about before. Each one of these cells codes for a small amount of a large problem of the map, and they do that in a distributed way. And so this grid cell here is going to be firing at regular periods across the map, and what it's coding is the definition of space, essentially how you get from one place to another in space, a relationship. So yeah, I guess everyone knows what these look like. They have hexagonal firing patterns, and they have offset phases, and so together they form an explicit basis set of two-dimensional spatial relations, which tells you what it's like to have a two-dimensional world. And so we wonder, well, if they can encode, if they're coding a general abstract relational structure rather than anything particular about space, we wondered if they would form basis sets for other relational information, like I was explaining in the state space example above, or before. Okay, and this is the work by Alexandru Constantinescu and Jill O'Reilly. So if you're going to try and do this in humans, there's some good news, because these cells have an angle to physical space, which is basically consistent across all the grid cells. They have that, and there are also some cells, some of those grid cells, the conjunctive ones, that fire more often when you're running aligned with this hexagonal grid than they do when you're running not aligned to that hexagonal grid. And so that means there's some chance of seeing this signal in fMRI because the bulk overall activity is different depending on which direction you're running in a predictable way. There's 6 times when it should be high and 6 times when it should be low. And my friend and colleague Neil Burgess and his postdoc at the time, Christian Dollar, had this ridiculous and brilliant idea to go and see if you could find this kind of signal while subjects were wandering around a virtual reality environment inside the scanner. And so what the logic of the experiment goes like this. The subject is moving from one place to another, trying to find something that it can remember, and the subject has a direction of travel. And so what Neil and Christian did was amalgamate all of those directions of travel from the whole experiment and ask, well, is there some 6-fold periodicity of the fMRI bold signal as a function of that direction of travel? This is like an unusually precise prediction for fMRI. It's not something your mum or dad could have thought up. But nevertheless, amazingly, they found this 6-fold symmetry, this 6-fold modulation of the bold signal in the entorhinal cortex, exactly where those grid cells live. But equally amazingly or surprisingly, puzzlingly at the time, they found this activity in some other regions as well, also regions associated with memory and spatial reasoning, but not regions where grid cells have been recorded in rats. And so in the medial frontal cortex and the posterior cingulate cortex, amongst a couple of other regions. And so it's difficult to know quite what to make of this when you haven't got the cells, but rather amazingly, just 3 years later, Josh Jacobs recorded some grid cells in the frontal cortex of humans undergoing epileptic surgery. And so these grid cells have never been recorded outside of the hippocampal formation in rodents, but are present in frontal cortex in humans. And so this is now a really unusual thing, right? This is an fMRI experiment with a precise prediction that turns out to be right. This is cool. Okay, right. So anyway, so we thought, given that unusual set of circumstances, we would try and profit from it. So we designed. As you've seen so far, we spend most of our time doing these stimulus-reward learning tasks. But everyone that does these stimulus-reward learning tasks tend to work with stimuli that are fractals, and so they're completely unrelated to each other, as if you were walking around a world, and all of your stimuli that you're getting were orthogonal to each other, which is, of course, not true in real life. So we thought we would design some stimuli that are not, or that don't have that property, that can be arranged sensibly into a two-dimensional map. And these are these stimuli. They are birds, and that you can define them with a two-dimensional vector that defines the length of the legs and the length of their neck. And so those are, in fact, the two principal dimensions of birds. So you can move in this now bird space, this abstract space, just like you can move in real space, and you can play the same trick. So a trajectory in this bird space is like stretching a neck or stretching legs. That would be a 90-degree trajectory. The subjects can't see what's on the left of your screen. They just see what's on the right of your screen during the scan. You can have a 60-degree motion, which is moving the legs and the neck at the same time. And so you can imagine that you can design schemes with these birds morphing and warping around that are just like the trajectories of movement in a real two-dimensional space. And so Alexa did this, and then it did exactly the same as what Neil and Christian had done, and looked for this six-fold modulation of the fMRI signal as a function of the direction of that movement. And so when you do that, you get something like this. You find these patterns of activity that are just almost indistinguishable from what you find in the spatial context. So here it looks like these, whatever is causing this six-fold symmetry, which we believe to be grid cells, is also causing six-fold symmetry when you navigate a completely non-spatial two-dimensional world, which suggests that those cells are coding something abstract about the relational structure of space rather than coding for the physical consequences of space. Cool, I might just skip these. I've got a bunch of things here to persuade you I'm right, but I've only got, is that 11 minutes till the end of questions?
No, till the end of the talk.
Oh, is it? Okay, fine, I can take you through it then.
No, it's 45 minutes total.
So it's 11 till the end of questions, yeah?
So in 1 minute it'll go yellow.
Okay, fine, I'll skip this then. That's what I thought. So these are just like, we did a bunch of stuff with this like split half analyses, calculating the grid angle in one and plotting the data in the other, and you can see the hexagonal pattern really clearly then. That's true in the VMPFC and the entorhinal cortex. Same thing works with a week between scans. So we got subjects back a week later, tried to do it really convincingly, and also tried to persuade you that the behavior relies on this pattern. The more of this grid-like pattern they have, the better they are at memorizing where they are in this abstract bird space. But I wanted to tell you a couple of other things. One is that we think that similar, for this argument to be right, if we're arguing that these structures are about the relationship between anything rather than just the particular relationship of space, then it should also be true for discrete things with discrete relationships as well as for continuous things. And so we think that that's true. So here's an experiment where we tried to test that. So this is an experiment where subjects were walking around a graph. They were walking around this graph by just answering irrelevant questions about stimuli that happened to be presented in an order that respected this graph. And so they would be asked, is this umbrella pointing to the left? Is this ring looking pretty? Or something like that. Well, they weren't. They were always asked left-right questions. But they were irrelevant questions, but they were presented those questions in an order that respected a graph. Just like if you were walking around space and you were presented with interesting plants to look at as you're walking around space, there might be some encoding of the physical space in the hippocampal entorhinal system, and here we hope that there's some encoding of the graph in the hippocampal entorhinal system. And indeed there is. If you look for parts anywhere in the brain where the similarity between voxel representations looks as if it respects the distances on the graph, you find it in the entorhinal cortex, just where you found those grid cells. And you can see, so you can see here distance 1, distance 2, distance 3 have, actually I'm plotting distance rather than similarity, so inverse similarity, have increasing distance. And then you can see this, so if you, we only measured this for 7 elements on the graph because of the various technicalities about measuring the way you measure distances, but you can plot these 7 elements in this distance matrix, and then so now you have a matrix showing you how similar or dissimilar the representation is for each of these 7 elements, and you can just do some dimensionality reduction and multiple dimensional scaling of that 7-dimensional thing, and you can recover that map, which is basically the exact graph that the stimuli were presented on. And the subjects were not aware that these relationships were present because they were answering simple, unrelated questions about the left-right pointedness of the stimuli. Cool. So it looks like those explicit relational structures can code for either continuous things or discrete things in the hippocampal formation. I would tell you something about the relationship between the OFC and the hippocampal formation if I had another 10 minutes, but I don't. So I'm just going to show you this last thing to attract you to poster 212 tonight, because this is something I'm really excited about that I'm doing with James Butler, who's in the audience, and Steve Kennelly, which is building these big abstract relational graphs into a monkey task. And so here's a monkey playing in his home cage on the excellent iPad system that James has built, and he's playing these navigation games on trying to get a reward, he's being told, go and find the handbag, go and find the bicycle, and he's having to navigate around these maps. And so here's one of those maps, and so you can see here's a 3-step navigation between, he's asked, go and find that satchel from this starting point of this electric guitar thing, and he has to make the right choices. And you can see he's really good at this, and so in these 3-step problems, he learns how to do all of them on the map within 5 or 6 days now. Cool, and there's some there's some early signs that he's learning that they've got this structure learning property, because as they encounter new problems on the map they've never seen before, they're faster and faster within each map, but also as they acquire new maps, because remember they're learning these maps really fast now, as they acquire new maps, they're faster and faster to learn those new maps. So go see poster 212 tonight if you want to see the monkeys. Not they have time, I hope the actual monkeys won't be there. Okay, I'm going to skip the OFC and why the OFC and the hippocampus talk to each other. And I'm going to end with this and just tell you that I think that we have these explicit relational structures in the frontal cortex, and these can control how new stimuli are processed, and that if you're going to represent relationships explicit, you better do it globally, not locally, and there's some analogies with space that I tried to point out. Cool, okay.
Tim?
Hi, Tim. Thanks for that talk. I had a question about what your thoughts are on whether or not our priors about the global structure of the world help us generalize or are inhibitory to this. So, you know, the fact that certain objects, you know, sort of come in sequence in our natural experience, or that we can tell stories like we walked by the tree, the tiger jumped out, the sun set because I was dead because it ate me. You know, do these sort of cognitive processes and priors, do you think they affect the generalization of the global structure that you find across tasks in these very abstract settings?
Yes. So I think that everything we're doing is about making rich priors for topologies, and I think I think there are all sorts of rich priors for topologies that we learn as a youngster. My son currently is making long lines of cars, and it's all he does. And he's building up the topology of line, which he's going to use later to be good at maths, I'm sure. And like I absolutely think that, and almost everything we're doing in our lab is trying to prove that now. Yeah.
Thank you. It was a great talk. So it wasn't clear to me from that last discrete task where there wasn't this hexagonal symmetry in the implicit task. You know, this one where you're moving around on a rectangular lattice rather than a hexagonal one. Is the representation changed in the same way, or is it also somehow mapped?
We don't know.
Okay.
That question. I think we will know within a year, because we've designed several fMRI tasks to answer it, and when we get an ambiguous result from those, we've also got a monkey task that's going to ask that we're going to go record. So I feel like we're going to know the answer to that question within a year, but we don't know it right now.
Thank you.
Okay, so I have 2 questions. So the first one is about the abstract graph representation. So when instead of a 2D space, use a complex graph with objects that one lead to another, I wonder if you can test the same kind of prediction from the Starkenfeld paper. So basically you would expect that your global representation is what you get from spectral graph theory. So you would have your successful representation, and then you should find the eigenvalues of that. So
I defer to Kim on almost everything, but I disagree with her on this point. I don't think we'll see, and we're trying experiments to test this. But I'm guessing that the grid cells will not be the eigenspectrum of your particular graph you're navigating right now. I think there'll be the prior distribution over the likely eigenspectras that you might encompass in your world so that you can rapidly construct new maps. That's what I think is going to happen. And so I think that you won't go and change, you won't be able to learn a new map and immediately recompute its eigenspectrum. That's my guess, but I'm hoping we'll find that out. Yeah.
Okay, thank you. And the second question was about the orbitofrontal cortex. So you mentioned that it was crucial to learn the structure correctly.
Well,
of the sequence.
I didn't demonstrate. I think it is, but I demonstrated that it was crucial after the structure was learned to be able to apply that structure correctly, because the lesions were made after the structure was learned. Yeah.
And I'm curious how it linked to the second part, so the part you were mentioning about orbitofrontal cortex and hippocampus.
So, yeah, I would have told you if I had 10 more minutes. I'll tell you later. It's fine.
Thanks a lot.
I mean, basically, I think that the OFC representations are really important for learning those models and for pointing to the right place in those models. It takes more than a second to explain this point, so I'll tell you later.
Hi, thanks for the nice talk. So my question is, well, actually, you found this very interesting representation in the frontal and entorhinal, and I'm curious if there are actually any differences between like MPFC-ish regions and entorhinal-ish regions that you've found.
So there are lots of differences that are definitely true between those.
in your task.
So there are no differences in the stretchy bird's task, but you might have a hint of the answer to this question just from looking at the second task I showed you, because there was very little frontal cortex in the second task, which was a passively performed task, right? The discrete task, right?
Oh, I thought there was like a
There was a
VMPFC-ish
tiny,
slash
yeah,
MPFC-ish.
yeah, but it was much smaller than the one in the, and so routinely in all the experiments we do, if you do something passive, you're no problem with detecting hippocampal stuff, never detect frontal stuff. If you're then navigating in that world, you get loads of frontal stuff going on. There are other answers I could give you as well, but that's a good start.
Yeah.
Okay, we have time for one more question.
Ida?
Thanks. Thanks for a fascinating talk. I was interested in the time course. Well, how long did it take for people to learn the bird task?
Quite a long time.
Did you track the development of the representations over that time?
No, we didn't, because we can't put people in the scanners for 4 hours. I mean, it took 3 or 4 hours to really learn it well. We were very careful because we wanted to make sure they had a precision of less than 15 degrees to make sure we didn't get Nyquist problems with our analysis. And so they had to really learn it really, really well. And so they were doing it for 4 hours. There's suggestive evidence from another study that's been done in Oxford by Matthew Rushworth's group that it might take, that you don't necessarily have that immediately, and it might take a while for you to make the analogy, essentially, with space. But it's really speculative stuff.
Thank you.
Thank you, Tim.